Forensic scientists have used hair samples to help identify the species, sex, and race of a potential suspect in a crime scene. The problem is, DNA can't survive when it's not in living cells. When hair grows, the cells stretch and die off because they're too far away from their nutrient supply. But what if I told you that proteins in your hair could positively identify you 98.3% of the time? Recently, researchers at Lawrence Livermore National Laboratory in California demonstrated how to extract genetic information from proteins in hair. The team consisted of Brad Hart, Clandon Parker, and Dion Annex, shown here. Mutations in DNA get translated to proteins, and sometimes they can change the protein structure. By looking at those changes in proteins, they can infer what was in the original DNA. It all starts with the DNA double helix being pried open by the RNA polymerase, which enables RNA nucleotides to pair with the template strand of DNA. A pre-messenger RNA strand is formed, but still has to go through multiple steps before being translated to proteins. The strand receives a 5' cap, which is 20 to 40 guanine nucleotides, and the 3' end receives a poly A tail, which is 50 to 250 adenine nucleotides. Large portions of the RNA molecule, called introns, are cut out from the RNA during a process called RNA splicing. Particles called small nuclear ribonucleoproteins, or SNRPs for short, recognize the splice sites. SNRPs join with other proteins to become a spliceosome. The spliceosome interacts with the introns and releases them, leaving the exons. Next, the mature messenger RNA is fed through a ribosome, so polypeptides can be formed. The first step is codon recognition. The anticodon of a transfer RNA pairs with a codon of the messenger RNA. tRNAs move through 3 binding spots. First, the A site, where the tRNA is held carrying the next amino acid being added to the chain. Next, the P site, which holds the tRNA carrying the growing polypeptide chain. Finally, the E site, where discharged tRNAs leave the ribosome. Once a ribosome is far enough past the start codon, a second ribosome can attach the messenger RNA, eventually resulting in a number of ribosomes along the messenger RNA, called polyribosomes. Often, there are changes called mutations in the DNA double helix that can change the protein structure. Insertions and deletions are types of mutations that have a large effect on the resulting protein. An insertion mutation is the addition of one or more nucleotide pairs, which will lead to improperly grouped codons. Using this information, the team assembled an experiment to test the accuracy of the proteins. They analyzed hair from 76 men and women, also 6 sets of skeletal remains from the 1700s and 1800s. Specifically, the team was looking for changes in protein structure caused by mutations previously mentioned. Those changes will help them figure out what was in the original DNA. They found 185 patterns of amino acids called markers to allow them to trace people's identities. They compared their results to the subject's DNA from blood samples and were correct 98.3% of the time. The team wants to broaden their research and test proteins from bones, teeth, and skin. Their overall goal is to make protein identification precise enough so that it can be used in the court of law, just like DNA.